Hello, welcome to Biostudy Pathshala. I am Nilanjana Sharkar, wishing you a very safe and happy day ahead, and also hope that you are leading a safe and healthy lifestyle in this time. So let's get back to today's class. So today we will do an introductory class on developmental biology, as because it is part 1 of the developmental biology, and today we will discuss about basic developmental biology and stem cell concept. So the developmental biology, it is the branch of the biological science which deals with the developmental procedure in any living organisms. So what is the development means? Development is a complex phenomena which is the formation of the complex cellular structure from a single and simple cells, and it starts from a simple cell and ends to multiple cellular structure. As this procedure, that means the developmental procedure, starts from a single cell and ends up to a complex cellular structure, that is why this is known as epigenesis, and it is a progressive procedure. It starts from zygote, and zygote is a haploid structure. Zygote is in diploid nature, and this zygote formed by the fusion of sperm and ovum. Sperm is male gamete while ovum is female gamete, and gamete means they are haploid in nature, and from this zygote the entire living organisms can be obtained. So the entire developmental procedure have some overlapping and sequential steps. So these steps are: first one is growth, second one is division, third differentiation, fourth pattern formation, and fifth one is morphogenesis. Overlapping and sequential means, if we assume about this figure, this 5 circles, this one is first, second, third, fourth and fifth. This portion is overlapping in between first and second circle. This portion is again overlapping in between second and third. So some portions are overlapping among these circles. So the developmental procedure also contains some basic overlapping and sequential procedures. These are, as I have mentioned, that growth, division, differentiation, pattern formation and morphogenesis. So what is growth? Growth is the increase in cell size. If we assume that this is a cellular structure and it gains its size about this much, it is known as growth. That means increase in cell size. Now second one is division. So division means the increase in cell number. So the division procedure is, from this cell, if 2 cells forms, this is known as division. So this division can be either symmetric division or asymmetric division. Symmetric division is if this cell divides into 2 cells and these 2 cells are identical in structure. This is known as symmetric division. And again, if this cell divides to form 2 cells, one is almost like this and second one is too teeny, or their structural or functional or physiological difference can be observed, this kind of division is known as asymmetric division. And third one is differentiation. Differentiation means diversification of cell types. So if we assume this is a cell and it further forms this kind of structural cell type, and its function also differs from a cell. This is known as differentiation. That means to gain the fate. Pattern formation is the organization of pattern. That means the differentiated cells can be organized in a definite pattern. And fifth one is morphogenesis. That means that differentiated cells gain its shape and size to gain its final fate. So next is the concept of stem cell. We are all aware of this term, that is the stem cell. Stem cell can be found in animal body system, and it acts as the reservoir of undifferentiated cell mass in our body. Reservoir means, if we assume that this is a reservoir of stem cell, within this boundary, this stem cell remains in undifferentiated nature, and once this stem cell leaves this boundary, the stem cell cannot be in its undifferentiated nature, and it will gain a differentiated or a final fate. This fate might be either epithelial cells or gastrointestinal cells or nervous system cells. Several kinds of cells might be. So what is the stem cell niche? This is the boundary within which the stem cells retain its undifferentiated nature is known as the stem cell niche. That means this the portion is known as stem cell niche, and here the stem cells can be in undifferentiated status, and stem cell can divide either asymmetric way or in symmetrical way. As I have discussed earlier, that what is asymmetric division or what is symmetric division. Asymmetric division, as you can see in this figure, this is a asymmetric division fashion, as because it is a stem cell, and upon division a cell formed exactly like stem cell and another cell formed as a differentiated cell or which differs from stem cell entirely. So this is known as asymmetric division. If we assume this stem cell, if this stem cell divide next and forms these two identical cell types exactly like its parent cell type, it should be known as symmetric cell division. Although stem cell divides almost or generally asymmetric fashion, but sometimes they can divide also in symmetrical way. So stem cell shows potency level. So what is the potency? Potency or the potential means the ability to do something. So what kind of work or what kind of act can be performed by a stem cell or by any stem cell? If a stem cell can divide to form any kind of cell types and also form total body or the entire body of any living organisms, mainly animals, is known as totipotency. Here toti means total and potency means the ability. That means the cells which shows the total ability to build the entire body of any living organisms, this kind of potential or this kind of potency is known as totipotency, and this totipotency can be observed or can be shown by zygote. Only the zygote have this kind of ability to perform or to build an entire body of any living organisms. Next is pluripotency. Pluri means not total but maximum. Here stem cell or the cells can form almost whole body but not the total. That means we can say that pluripotency is narrower than the totipotency, and this pluripotency can be shown by ICM. ICM means the inner cell mass, and it can form the whole body of fetus but not the trophoblast. Trophoblast further can form the placenta of any fetus, and this trophoblast cannot be formed by the ICM. Next is multipotency. It is further narrower than the pluripotency, and this kind of cell can make multiple cells but not like the ICM. For example, HSC, that means the hematopoietic stem cells. Hematopoietic stem cells can form the blood cells, for example RBC, WBC, platelets, but they cannot form many other cells in any living organism's body. Next is oligopotency. If a cell can perform a potential to build up 2 to 3 kind of cell types, this is known as oligopotency. That means it is again smaller potency than the multipotency, and this kind of potency can be shown by either lymphoid and myeloid cells. Next is unipotency. Uni means one. If cell shows one type of potency, that means one cell can form only one kind of final cell type, that is known as unipotency, and this kind of potency observed in case of spermatogonia and oogonia, as because spermatogonia forms further sperm cells or sperms and oogonia further forms ovum. So if we arrange this potency level in a descending order, so first will be totipotency, as because it is the total potency of any cell to form the entire body. Next is the pluripotency, as because here cells can form whole body but except particular one. In this case ICM performs pluripotency nature, as because ICM can form whole the body of the fetus but not the trophoblast. And the example of the totipotency is zygote. After pluripotency, next is multipotency. The example of the multipotency is HSC, that is the hematopoietic stem cells, as because this kind of stem cells can form all kind of blood cells but not other kind of body cells in any living organisms. Next is oligopotency. Here the cells can form 2 to 3 kinds of cell types, for example lymphoid or myeloid cell types. And the last one is the unipotency, as because here the cells can show the potential or the ability to form only one kind of cell types, that is the spermatogonia or oogonia performs like this way, as because they can form either sperms or ovums. So as you can see in this figure as well, that this yellow portion is trophoblast and the inner portion is known as ICM. And this blue region is ICM, and this ICM can build total or the entire body but not the outer boundary, that is the trophoblast, and this trophoblast is very much essential for development of any fetus, as because it provides the placenta. Totipotent is morula, and this morula or this zygote can be formed by the fusion of ovum and sperm. So next is types of stem cell. Stem cells mainly of three types. These are embryonic stem cells, fetal stem cells and adult stem cell. Embryonic stem cells, this kind of cell types or cells can be found in case of embryonic stage, and this kind of cells either be totipotent in nature or pluripotent in nature. This should be pluripotent, not multipotent. Totipotent means if the embryonic stage is zygote, and pluripotent means if the cell is ICM, and these two shows embryonic stem cells. Next is fetal stem cells. These stem cells can either be pluripotent in nature or multipotent in nature, but cannot be totipotent in nature, as because the embryo grows to fetus, and fetus formed by only the ICM, not by the trophoblast. So the fetus can show either pluripotent kind of potency of the stem cells or multipotency of the stem cells. Next is adult stem cells. This cannot be either totipotent or pluripotent. It can be only either multipotent or oligopotent or unipotent, as because adult stem cells, for example HSC, that is the hematopoietic stem cell, which shows multipotency. Oligopotency can be shown by either myeloid or lymphoid, and the unipotency, which is shown by the gametic mother cells, which are spermatogonia and oogonia. So as you can see in this figure as well, the totipotent embryonic stem cells giving rise the pluripotency kind of embryonic stem cells, and further this kind of pluripotency results in the formation of the multipotent kind of stem cells further. So stem cells, if we differentiate these embryonic stem cells and adult stem cell, embryonic stem cells derived from the ICM, and it shows pluripotency in nature, and it can make whole organism but not the trophoblast. And adult stem cell can be found in adult body cells and tissues as well, and it shows either unipotency or multipotency or oligopotency kind of nature, and this kind of potency either observed in case of gamete formation, or in case of blood cell formation, or in case of repairing or replacing in any damaged cells or tissue. Here whole organisms cannot be made by this kind of stem cells. So this is the figure of stem cell niche. Here some factors act in this niche to retain the undifferentiated nature of the stem cell, and if any of the stem cell leaves this niche, these factors cannot be reached up to this kind of cells, and these cells cannot retain its undifferentiated kind of nature, and it gains its fate, and that is why it is known as specified or sometimes differentiated cells. And in this stem cell niche, those factors which can retain the uncommitted or undifferentiated nature of the stem cells is known as paracrine factors, and these factors can act within small range, mainly in the stem cell niche, sometimes in other portions we will see in our next classes as well. But if any cell leaves this stem cell niche, the paracrine factors cannot be reachable up to this kind of cells, and this kind of cells can gain its final fate and not again regain its undifferentiated state. So in this flowchart this is undifferentiated cell types, and as you can see that color gradient, this is almost a lighter color. And if we suppose that this is a cell which is stem cell in nature or undifferentiated in nature, if it is specified, it steps forward to its next step, and in this stage this A cell can be reverted to its uncommitted or undifferentiated cell from this specification or the specified type of cell. Next, it further step forward towards the determination, and once a cell get determined, the cell cannot be get backward to its undifferentiated in nature, and once a cell is determined to its final fate, the cell progresses its fate towards the differentiation and gets differentiated and form differentiated kind of cell types. As you can see that this straight line arrow pointer shows the gaining of fate of any undifferentiated cells. It is one step forward, next step, third step and fourth step, from which the differentiated cells cannot be stepped back towards the undifferentiated cell types. And this kind of arrow is showing that the probability of any cells to get back to its undifferentiated kind of nature. This is much more darker arrow which confirmed that from this state a cell shows maximum chances to get back to its undifferentiated in nature, and from this state the cells show less chance to get back to undifferentiated nature, and from this it lowers more, and in this step once the cells are determined, the cells cannot show any steps to get back to its undifferentiated nature. So this is for today's class. Thank you guys, and if you find out that this approach is helpful for you, you can give a feedback and you can like this video. And if you like this video, you can share this video with your family, friends, senior, juniors, and also subscribe this channel. And also don't forget to maintain a healthy and hygienic lifestyle. Until then, be safe and be healthy. Bye bye. We will catch up after a few days. Thank you once again.